Good morning. Thank you for joining us for our latest Department of Defense Information Analysis Center webinar titled DNA as a Molecular Engineering Platform for Defense Applications. My name is Steve Rutherford, and I have the privilege of being the director of the HDIAC, and today we are hosting Dr. Steve Armentrout for this presentation. First, as we usually do, I want to start off by thanking all of you and saying that I'm grateful to everyone who's taking time out to join us during these tough times. I'd also like to thank our sponsor, the Defense Technical Information Center. The DTIC-IAC mission is to collect, synthesize, produce, and disseminate scientific and technical information to DoD and other government users. If you've not checked out DTIC's website, you can find it at www.discover.dtic.mil, and I encourage you to take a moment to review their expansive services. And also, without DTIC's sponsorship, this webinar series just wouldn't be possible. Also, a few admin notes as we kick it off today. Please note that all the attendee lines have been muted, so if you have questions during the webinar, we're going to ask you to submit them using the attendee chat box you can see on the left-hand side of the screen. We're going to work to save the last 10 minutes or so of the presentation to go over those questions and discuss them. Please also note that this webinar is being recorded, and a link to the recording as well as the slides will be available at the HDIAC website for later download. And with that out of the way, it is my pleasure to introduce you to today's presenter, Dr. Steve Armentrout. Dr. Armentrout is co-founder and CEO of Parabon Nanolabs, a DNA technology company named in 2018 by Inc. magazine in its top 5 most innovative companies. Dr. Armentrout is a machine learning expert with nearly 30 years experience designing and using high-performance computers to solve challenging problems in a wide range of domains, including stock selection, facial recognition, and DNA nanocarrier design. For the first 15 years of his career, Dr. Armentrout and his colleagues tackled computationally demanding problems in domains ranging from neural modeling to investment forecasting. He's also served as principal investigator for research projects funded by a variety of organizations to include NASA, DoD, DOE, and the NIH, just to name a few. Under his leadership, Parabon advanced forensic DNA analysis with its Advanced DNA Analysis Service, which began as a DoD contract with the original intent of identifying bomb makers who had created IEDs. Since being made available to law enforcement, the technology has generated leads resulting in over 130 positive identifications in cases that have been cold on average for over 25 years. Dr. Armentrout holds a PhD in computer science, and he specializes in neural networks holds a PhD in computer science, specializing in neural networks from the University of Maryland. And with that, I will turn it over to you, Steve.
Good morning, everybody. Most of that introduction was about a part of our company that analyzes DNA, and indeed that's what we are best known for. But this morning, I'm going to talk about a division in the company and what we do on the other side, which is building things out of DNA. And so hopefully you take away from the presentation a new appreciation for what DNA can do other than be a biological molecule for storing information. I need to find my opportunity to advance the slides, and after that, we'll be good to go. So previously, I had an advance button that's no longer present. Okay, now we're good to go. I figured we would start off with some basics about DNA so that everybody was on the same page. And we're all familiar with DNA as a biological molecule, that is, a helical molecule comprised of 2 strands that run anti-parallel with one another. They're held together by hydrogen bonds between nucleobases, and we call this base pairing. And so you've probably seen this in lots of different representation. This is a single bead per nucleotide representation. And the bases are adenine, thymine, cytosine, and guanine. And as you probably know, A's bind to T's and C's bind to G's. We'll go to the next slide and look at a different representation that makes that more clear. And we call segments along the DNA that have this complementarity reverse complements of one another. And we sort of take this for granted because we always see DNA represented as a double-stranded molecule. We always see it in the biological context, and biology has figured out that this is a wonderfully stable molecule and it can be used to hold information. And so biology has all these tricks for being able to cut DNA, repair DNA, do parity checking. Biology has really exploited this double-stranded complementary DNA. But in fact, if you talk to a chemist, they think of DNA a little differently. They think of DNA as a single-stranded polymer, which in fact it is. And so here it is in its more native form, as a single strand that may have some self-complementarity, may not. We've shown some here just to sort of show how a hairpin might form along these four bases that are reverse complements of one another. But importantly, we have control as engineers of the sequences that go into strands. And I didn't know this when I got into the field 15 years ago, but it's commonplace. You can go out and order a strand of a specific sequence and have it shipped to your front door in a couple of days. So immediately we have this interesting material. It has this complementarity property. We know how to synthesize it. We can synthesize it either in the small, down below 50 bases, but today you can even get synthesized DNA at the gene scale up to 5,000 bases, and even more if you're willing to pay up. So we have this molecule that has the hints of being programmable, because we know this complementarity tends to attract segments that are reverse complements of one another, and one begins to ask the question, can you take advantage of that? So before we go there, I thought just to sort of underscore that DNA is a single strand that is stable when you have complementarity. We show this video of duplex hybridization. So what we're going to see is two single strands using a simulator called OxDNA, and I have the full citation down below. And I think Steve will kick off this video for us now, and you can watch two single strands in a simulated box, eventually sampling one another. And because they're complementary, they will eventually form a stable duplex. And this is only possible because of that complementarity. If these strands did not have that complementarity, they would sample one another, but then wander off. What we see here is a wonderfully stable molecule that is being held together by hydrogen bonds. And what happens in biology is those hydrogen bonds will run for great distances and store information. And so one asks the question, well, okay, if we can make some sections bind where we want them to, can we in fact create structure? And back in the mid-'80s, Ned Seaman at New York University hypothesized that that was possible. He hypothesized that you could form branch junctions of DNA, and if you got the sequences right, they would form different structures. So at a glance, what we see on this slide are two helices stacked on top of one another. But if you look more closely, you can see that the blue strand on the left is actually participating in both helices, as is the lavender strand on the right, and in fact the yellow strand as well. So it's only the red strand going across the top and the green strand going across the bottom that are singularly participated in helices. And so Ned's insight was that these junctions where you can cross over from one helix to another could be programmed in a way that was non-biological. And you could immobilize those junctions with the right set of sequences and begin to weave DNA. So this was hypothesized, as I said, in the mid-'80s. It was later shown to be true and possible. And by the 1999, Eric Freeman's paper, showed ribbons of DNA being able to be built this way. So this was sort of the beginning of what was called structural DNA nanotechnology, for which I predict Ned will someday get the Nobel Prize. In fact, he was shortlisted not long ago. So once you have the ability to think about DNA as a weaving material, you can then use it to self-assemble. So as we saw in the video, those two strands sampled one another because they were complementary. They found a way to line up in a stable form, and they did that by themselves. So if we have the ability to control complementarity, and we have this inherent self-assembly mechanism, then you can use self-assembly to create more complex structures. So what we're going to see next is another video, this time where we're taking advantage of those crossovers, and we're going to see a simulation of what's called origami, and I'll describe it in more detail. But what we have here are single strands of DNA that have complementarity in certain places that has been pre-designed, and they sample one another and find where that complementarity works. Where it doesn't, they bounce away. And although this happens a little more, well, of course, rapidly, but with a little more collision in real life, what you saw there was the formation of a pre-designed structure using DNA. And the term origami has become popular to describe designs where you start with a big circular scaffold of DNA and then smaller staples that go in and fold that longer scaffold into place. So already, I hope you appreciate that the DNA is a programmable material, and we can use that to make very sophisticated structures and make lots of them because it's self-assembling. So many of these designs that I'll show you today are one-pot syntheses that create trillions of copies of the same structure that's there by design. So what we have here at the top is just a really simple illustration of 9 strands designed to complement in a specific way to create this plus sign motif. And with a little trickery, we can leave some of the ends of those tiles unbase paired, complement them by design with other tiles made similarly to create the 4x4 structure that you see. And those 4x4s can be also woven to create 8x8s and so on. So this is just a nice simple example of a compound structure created by properly ordered DNA sequences. Down below is just a variety of different DNA nanostructures created similarly. And then on the bottom right, our team is showing off a little bit by creating oligos with streptavidin at the top. And that streptavidin, when you do atomic force microscopy, has a greater Z height, so it shows up brighter, and they've written the letters DNA. Just to give you some sense of the precision of placement that we have with this technology. With partial funding from the Department of Defense, over the last even more than 10 years, we've been working on a piece of software called InSequio. And think of it as CAD software for creating DNA nanostructures. And what we see here is this idea that everyone wants in CAD, and that is the ability to go from a concept, a napkin drawing, to a CAD rendering. And what you want is a tool to help you organize not only your design work, but the creation of the sequences that are required. I didn't get into it earlier, but think about it. Because you have complementarity, it's not enough just to program where you want the complements to be, but you want to avoid accidental non-specific binding because that could foil your design. But you want software to help you with these processes so that you can order up the right strands, do the synthesis, and using self-assembly, get the design that was intended. We have here a couple of videos in succession that will show some of just the core capabilities of Insequio. The first video is just showing the basics of manual manipulation. So now, think of this as a CAD tool, sort of a sophisticated 3D version of PowerPoint, if you want to think of it that way, that's specialized in DNA nanotechnology. So what we see here is we've drawn a duplex, we've nicked a particular site, and then we'll go back to that site just for illustration and show how easy it is to splice those back together. And we'll double click on this and show that the strand is in fact intact again. We're going to draw now as another strand of DNA and show you how easy it is to create a crossover. And so you could imagine if you had a particular design in mind, using a drawing CAD tool like this to come in and systematically create the design and then ultimately create the crossover locations that hold it together. You can imagine, these two strands, these two duplexes by themselves in solution are going to be all over the place. But if we introduce double crossovers, it's like weaving textiles together, and that forms the stability. So here we go. We're going to move this just a little bit. And once we have it lined up, then we can use the tool to specify a particular location that we'd like to introduce a crossover, a few clicks to introduce the bases or to identify the bases that'll be participating in the crossover. And then with the right mouse, we'll hit from the pop-up menu to create the double crossover. Didn't quite show there on my screen, but what happened was we created the double crossover, and then we went to a color palette to color these new strands that were created by the crossover operation. So this is something that's not easy to do by hand. You want software to take care of these steps for you. And then just as importantly, sometimes during a design, you say, okay, I put a crossover in a location, I don't like it there. I need to be able to remove that quickly and simply. And that's what we've done here. You can see that that strand now is highlighted and contiguous as it was before. We'll do the same coloring on the top one just to round out that piece of the demonstration. So this video was just really elementary things that we need to be able to do to build up a DNA nanostructure. If we go to the next slide, particularly because I'm a programmer at heart, this is very exciting. It's scriptable. We've created a Python API to Insequio, and what you see here is some Python code that exercises that API to demonstrate the capability and draw inside the Insequio window. So I'm using Microsoft VS Code here, and we have this program, and it can talk across the network to an instance of Insequio, which it's doing here. And I wish I had a little circus music to round this out, but you get the basic idea. We're just using this tool to show the power of the scripting language. And once you can begin scripting designs, now you're outside of the realm of doing small origami that you need to go in and do by hand. Now you can use software to create sophisticated designs and retool them over time, parameterize them, for example, so that you can change their size and shape. And this really brings power to the platform. So we'll rotate this around just so you can appreciate the three-dimensionality of it. I don't know about you, but I find it challenging to work in a 3D CAD application. We have support for construction planes and doing all of this stuff in the editor, but doing it via script I find easier. Here we're showing on the right that we've got a strand panel to help keep track of the strands, the sequences that comprise them, and you can select a strand on the right and it highlights in the left is what we're showing. And on the upper corner here is a properties menu, so we can select specific pieces of the structure, whether they're bases or strands, and they'll show the properties in the right, and we can edit those properties as well. Finally, we're just doing a little spin here to round out the demonstration. And what I hope you took away was the fact that with a Python API, one can begin to create arbitrary designs in the Insequio platform, and then using all the tools that are already there, you can go in and do manual manipulation. And in fact, this is how we work today. We do a lot of our design with scripts, and then we come back in and do manual manipulation for fine tuning. If we go to the last video, we're going to see a larger structure this time. So I think not sure what's happening here on my screen. There we go. I see it now. What you should see is a hemisphere that was designed with scripts driving Insequio. And this hemisphere is intentionally not perfectly hemispherical. We wanted to use up all of the plasmid scaffolding that we had available to us. People tend to use the M13 plasmid for scaffolding. It's inexpensive, easy to get, but it has a fixed number of bases. So when you create a design, if you have extra bases, they tend to hang outside of your design. We designed this one so that there wasn't anything hanging out. But what we're showing here is, I think, really cool. We've got a 2D view and a 3D view. And although drawing in 3D doesn't automatically create 2D, we're working on that kind of capability. We can write our scripts such that they produce both at the same time. And what that lets you do is synchronously work on one portion or one view of the design while you're also seeing the updates on the left, on the other part of the design. So here, what we've done is select a strand in the 2D schematic view on the left, and then it shows it highlighted on the 3D view in the right. And we can use, our fit selected capability to zoom in on those specific locations. So I think the future of the tool is really to make the 2D to 3D transition as seamless as possible. We know that that's impossible in the general case, but the idea is to create tools so that users can very quickly go from a 2D to a 3D design after they've made changes or de novo. So that's just an overview of Insequio. We're really appreciative of the various DoD sponsors and NIH and NSF sponsors over the years that helped us enhance this tool. Next, we'll move on to Insequio VR. And it's unfortunate, VR doesn't really come through too well in a webinar presentation. It's one of those things you really have to experience, but as fond as I am of using CAD to do this kind of work, I have to say that it's not always easy and intuitive. Whereas with VR, because you're immersed in a 3D environment, you can manipulate things readily in 3D in a much more intuitive and quick way. And so we're extending the Insequio interface to support the Oculus platform. And what we see here is a mock-up of what a user might see, but then here's an actual design and an AFM on the right of that design. And then as best we could, we got some screen grabs just to sort of illustrate what it looks like in the VR environment. Again, there's no way you can fully appreciate this without actually having the goggles on, but what we have here is a tube on the left, and one can imagine that you can shrink down in size and zoom right through the middle of this tube, or on the right-hand side, you can scale it down such that it fits in the palm of your hand, scale it up to the point where you're manipulating things, at the bead level with your hands or the controllers, as it were. So this is the kind of thing, as I said, you need to experience in person. If you ever get to the Reston, Virginia area, stop by our office and we'd be glad to give you a demo. Bring your own VR gear as long as the pandemic is running. All right, next we'll sort of ease into some of the uses and applications of the technology, I thought I would start with a simple design that has found a surprising number of uses. This is a cross-tile origami. Ned Siemens' lab, I think, was probably the first to produce such an origami, but there's been lots of variations on this design. It's about 100 nanometers across. Just for fun, recently, for a child's classroom, I did the math for them. How many of those things would it take to circumscribe a human hair? And I thought I would share those numbers with you now, just to appreciate the scale that we're operating on. You know, according to Wikipedia, the source of all truth, the diameter of the average human hair is 75 micrometers. And we see down here that 2,356 of these little cross-tile origamis would be needed to circumscribe something of that size. So, you know, if you're not thinking about the implications of working at the nanoscale every day, it's kind of fun to put it in context, just how small these are and the control that you have at that level because of self-assembly. So here are our AFM images of cross-tile origami produced by my colleague, Mike Norton. He's a professor of chemistry at Marshall University and also a co-founder of our company. He and another of our colleagues, Dr. Rahman, created these in 2014. And you can see the wonderful separation and precision that we have here with this design. And here's a close-up AFM image. Again, AFM is a tapping technology, and it's showing Z height, so things that are brighter are higher in Z height. And this is imaged on mica. And this is a recent paper where they're using these designs for super resolution microscopy. And so this is a really interesting technique to try to improve on the resolution of optical microscopy. And what we see here is truly the programmable nature of these designs. When you have the monomer, all of the fluorescent labels on this design at the tips are visible. And so if you look on panel A and then let your eye slide south to the middle panel, you can see all of the fluorophores lighting up. But when they form dimers, and they do that by design, when sticky ends are able to bind to their counterparts, then this stops the labels from being visible, and you can see in B in the middle row that they're no longer visible. And so that's really extraordinary control at this scale. And the vision is to be able to use this kind of technique for calibration. In fact, there's already commercial applications where they're using DNA nanotechnology for microscopy calibration. And again, I'm just showing you a few examples here to try to stimulate your thinking about what can be done with the technology. Next, we have artificial antibodies. And so this is a design from a previous DoD effort that we were involved in, where we used that cross-tile for some of the design, but on the right-hand side, we have a set of pincers that were designed using 6-helix bundle as a standard technology, or standard design, I should say, motif. And the idea was to create pincers that are not unlike the performance of an actual antibody, and then use that as a proof of concept to capture a particular target. And so I'll zoom up on these A and B panels with the next slide. And what you see on the left is an AFM image without the target streptavidin protein in the mix. And you can see that most of the pincers are in open position. And on the right-hand side, this is after the target protein was introduced and the species has been largely captured. I think most of these are, in fact, in the closed position. So it's a pretty nice demonstration of being able to use this technology in the context of nanosensing, nano-reporting. And so we think this application has a bright future. Next up, again, just trying to give a sense of the breadth of the technology. This is some work out of MIT. Yen Ping's lab and Mark Batta did this work. And they used the DNA to mold composites. So what we have here is the molding of silver nanoparticles and gold nanoparticles. And they used the DNA to plant a seed for either silver or gold. And then by introducing the appropriate reagents, they could grow that nanoparticle within the mold and then later take imaging of it as you see below. And they showed in this paper really exquisite control of the design, very nuanced resolution, if you will, of the molding capability. And so you can imagine all manner of molding application using the DNA, not as the primary structure, but to cast the mold, to be the mold itself. Again, just trying to show the breadth of where this technology can go. And this is a recent paper where, as opposed to molding, now we're talking about using the DNA really as scaffolding. And so in this paper, they applied 2 layers of scaffolding. First was a silica layer, and then next, a superconducting layer that formed lattices that could then form what they call Josephson junctions. And these authors were able to demonstrate that the resulting lattices had the performance characteristics consistent with Josephson junctions in superconducting. And here again, the DNA is really just serving to get the structure started, after which the scaffolding creates the actual effect that's desired. So we've seen applications where the DNA is being used as a mechanism for target capture. We've seen examples where it's a mold. Here we see it's just scaffolding. There are other applications that I've not shown here where people demonstrated the ability to create logic gates. There's a whole subdiscipline in the DNA nano field for actuation of structures. Again, not something I've shown here today, but hopefully you get a feel for the potential of DNA as a molecular engineering platform because of this versatility. And I'm closing here with a slide on work that we're involved in using the DNA as a nanocarrier. So you could imagine if you have this kind of control, being able to use the DNA to bring other moieties into position. So you can use oligochemistry to attach targeting ligands or therapeutic payloads, and through self-assembly, even though those molecules inherently wouldn't want to find the position that you want to put them in, the DNA can pull them into position. And that functionalization is a really powerful aspect of the platform. So we have several projects where we're using DNA as a delivery carrier. We have 2 with NIAID to develop vaccines against HIV. We recently got a fundable score from the National Cancer Institute to develop a novel treatment for prostate cancer. And we have a DoD-funded effort to use these same structures to create cognitive boosting agents. And there have not yet been any demonstrations in human, but there's a small and growing literature around the use of DNA nanocarriers simply because of their potential to program all these different moieties into a single macromolecule that can be used for therapeutics or vaccines. So with that, I'll say that I hope you have walked away from the presentation with an appreciation for the programmability of DNA, that it is something that, because of all of the investment going in around DNA for its gene synthesis work and so on, that it is a field very ripe for innovation because there's so much going on. This would be the 1960s equivalent of the graduate saying, plastics. I'm here to say DNA. I think DNA as a building material has a bright future and one that we're very excited about. So with that, I'll wrap up the presentation proper and open the floor to any questions you might have.
Steve, thank you very much for that fascinating presentation. I think it did spark a lot of interest from our viewers, so we'll just go ahead and dive right into the questions with the time that we have remaining. So Daryl Johnston was kind of curious about introducing an artificial antibody into the human system, and he was curious about how accepting an immune system would be of a DNA-based nano micromolecule. Are you aware of any work or any papers where anybody's looked at that?
Let's see, there's been a little bit of work on immune response, and I think it's a valid concern. We certainly know that CPG motifs will trigger immune response, and so these are things that would need to be guarded against. Our current concept of an artificial antibody is in vitro, but our nanocarriers are certainly intended to be in vivo. So there's a few tricks one can play. One important aspect of the designs is that you can do additional ligation after you created these things so that nucleases have a tougher time reading the DNA and consequently the immune response may not get an opportunity to be triggered. As well, you can coat the DNA nanocarriers with various additional layers, substrate layers, to try and slow down their exposure outside the cell. So one thing that I think is important to note is that while the work thus far has largely been using M13 because it's convenient and easy, when we get to human trial and so forth, we'll be able to design de novo sequences specifically to try and avoid any kind of immune response. But those are things that are down the road that we're going to have to address. In the animal studies that we've seen so far, and in our own animal studies, we've not seen any immune response of note. But obviously, more research needs to be done.
Super. Thank you very much. So Emily Pollard asked, she asks, does the development of unnatural base pairs for genetic code expansion provide new opportunities for DNA structures? And if that's the case, would you be willing to elaborate a bit on the ways that these unnatural base pairs advance the work now or could in the future?
I think there's an excellent opportunity there. There's a sort of a limited design space that one can explore when you only have 4 bases. So by introducing artificial bases, provided they perform similarly to the natural bases, then you've immediately reduced the challenge of sequence optimization. You haven't eliminated it, but you've given yourself a greater design space. So I think there's probably a natural marriage of the artificial base work that's being done with DNA nanotechnology. We've not yet worked with artificial bases, and so I can't say definitively that they will work as well, but it's certainly tempting to try and go there simply because of the extra design space that it gives you with sequences.
So I think that there's been lots of reports and lots of compliments on Insequio and the kind of the drag and drop CAD as to use layman's terms. And I'm sure you offer a lot of examples of how it's being used. So Are you, or what hurdles do you see to make this kind of DNA manipulation more common in manufacturing?
Let's see. So there's the challenge of scale up, and there's been some nice work over the past 3 years coming out of the Dietz lab in Germany, demonstrating biotech techniques for scaling up the amount of DNA materials that can be produced. So I see a good glide path there, work that originated from Chris Voigt out of MIT, and now in the Dietz lab. So they've shown the ability, to create gram scale quantities of the material at affordable prices. So I think that hurdle is slowly coming down. I think also, this is a field that is looking for the killer app. I feel a lot like, those of us who are in the field can probably sympathize with the 1960s, Fairchild semiconductor. I mean, they knew if you can control, electronics and logic gates in an integrated circuit, something good was going to happen. But I'm sure there was a period of time where they knew that, and yet they didn't know what the next killer app was going to be. And I sort of feel like that is where we are now. I mean, but that being said, we're seeing a rapid increase in the literature around the technology. And I think, once commercial applications really burst into existence that are demanding the DNA nanotech, then the manufacturability and the workflows along that will follow very rapidly. But right now, most of the work, is in research labs, and they're doing it at a scale that's affordable and appropriate for a research lab. Once the applications come along that demand manufacturability, I don't see that as a major problem at this point.
Okay. So Miguel Galindo asked about preparing large DNA structures. Specifically, he states that many short sequences have to be used when you create a large DNA structures, and he postulates there's a greater likelihood of unwanted sequences assembling. And he's curious about how do you engineer it so that the many DNA sequences will bind together yet minimize unwanted assemblies?
That's a great question. So we call that problem a sequence optimization problem. And it was one of the first challenges Mike Norton presented me when we began working together. We've developed software to do that. There are other tools out there. One thing to note is that when you're using what's called origami, we talked about, oftentimes you're given a sequence by the scaffold, right? If you use a plasmid from nature, it has a sequence. And really the only optimization you can perform is to sort of rotate that through design space to see whether the staples that are forced by that sequence accidentally mispair anywhere in your design. And so whether you're doing de novo sequence optimization, that is designing your scaffolding from scratch, or if you're using a plasmid and rotating it through design space, in either of those scenarios, you're looking for non-specific binding. You're looking for mismatches. You're looking for that. And basically you favor those sequence sets or permutations. In the case of origami, where you have a plasmid, you're looking for those that have the minimal number of mismatches. And it turns out that because of competition, longer runs of complementarity tend to win out over shorter runs. So as long as you get the mismatching down below a length threshold that is shorter than, let's say, the average length of the strands that you're trying to attach, the specific binding locations, if you can get your mismatches down below that level, they tend to get outcompeted by the longer strands. So that's not hand waving. It actually works out pretty well in practice that with conscientious but not insane amount of sequence optimization, the systems perform very well because of this sort of inherent ability for longer runs to outcompete shorter runs of complementarity.
So does Insequio have some sort of optimization engine associated with it?
Yes. Yes. So although I didn't show it today, we can take a design once it's complete, and let's say it's entirely full of wildcards. We can then, run a command that says go optimize, and it kicks it off to a server. Some of these optimizations take a long time, but then when the optimization has finished, it'll bring those sequences back, and you can say, please apply those to my design. Any other questions?
Yes. Gabrielle just asked a little bit about industry standards, and you were really at the forefront of this, been working on it for many years. What can you talk about engineering standards, or industry standards for things like designing for biochemical printing, et cetera, et cetera?
To my knowledge, I don't know that there have been any steps taken towards standards. I think it's probably a little early to contemplate that. If someone knows otherwise, please apprise me. But it's the kind of thing that I think will be application driven. Once applications come along that are demanding the technology is proving itself effective, then we can begin talking about standards for synthesis, standards for metrology, et cetera. But I'm not aware of any efforts yet along those lines.
So we had a question from, I'm going to massacre his last name, Thomas Solis, who asks, similar to the way that CAD has built-in block references, he's curious to know about what standard designs or DNA sequences would come within Insequio 3D, if anything.
We're working on what we call parameterized motifs and having a gallery of sort of the major workhorse designs that one sees in the literature. And that's, we all like to be able to do that. We like to be able to go into PowerPoint and choose a rectangle tool and draw with it and get a rectangle. So we understand that, and the scripting capability is giving us the means by which to create, a tool that says, give me a sheet of a certain dimension, and it takes care of everything, including crossovers. So those are in the works. We don't have that gallery yet, but that's absolutely the way to go, right? None of us want to have to do any of these things meticulously by hand. And so we're very keen on that exact idea. It's just, it takes work to get that gallery created, and we're working on it.
Okay, so another question from our live streams, has machine learning been used in consonance with your work to solve particular problems via the API? If not, could it be used in some fashion?
We've not used machine learning on the API, no. I think there are applications for machine learning. We do a lot of molecular modeling, molecular dynamic simulation on the DoD supercomputers, in fact, and that lets us, get basically an in silico microscope into these designs, but they're large, and using something like NAMD, you can run for a week on hundreds of core and still not get enough time, to make strong statistical conclusions. But I think there's an opportunity to combine supercomputing simulation with machine learning and improve on coarse grain simulation, predicting, likely structure. And we're seeing more and more of that. I think there was an announcement just this week about protein folding with the DeepMind team. So I think there's opportunities to do something like that in DNA Nano. We've not engaged in that yet, but you can bet I've thought about that more than once, because I think there's a rich opportunity.
So there's been lots of talk about what your technology can be used for and what the results of it can be used for. Things like biosensors. You gave a couple of examples. What are some other examples that you're aware of where this technology is being pursued?
I touched on it briefly. You know, there is a branch of the field where they're looking at electronics and trying to mimic what's happening in silico. I'm a little less, I'm sorry, with silicon-based computing, I'm a little less optimistic about those efforts, if only because the existing industry is so well established and can put so much money behind problems that for DNA Nano to make a real dent in that field, it's going to have to be, 100 times better. And those are hard things to achieve. But certainly, again, I only touched on it briefly, the actuation that's possible. So I didn't briefly described, I mentioned that long runs of complementarity can outcompete short runs. And so that can actually be used, they call it a toehold displacement, but basically I can introduce a new strand into an existing design, and if it has the right features, it might go in and displace a strand in that design, and the new strand might have a different property. It might have an overhang that's important. And there's lots of variations on this theme, but the point is now you can transition from state to state. And so having statewide changes because of changes in the environment or changes in the system because you've added ingredients, opens up the possibility for very interesting chemistries. And there's a subdomain of the field where they're looking at chain reactions, chemical chain reactions that can be formed, with the introduction of DNA strands. So it's a combination of, the structure provided by the DNA substrate with the moieties that it can hold and the displacement thereof. So you can see, glimpses of very complex chemical systems being created with this as a core technology. But I think, sensing and biological applications, I think, are the most likely big opportunities. I mentioned metrology in passing. I think there's already some early commercialization efforts there.
Well, Steve, thank you very much for that presentation. We got numerous compliments on it in the chat, so I'm really grateful for you taking time out for what I know is a breakneck schedule to present for us today. And for our audience that's out there, Parabon has presented for us in the past as well. We'll send out a reminder of Dr. Ellen Graytek's earlier presentation, which we talked a little bit about in Steve's biography when I started off today. But again, thank you very much. And thanks to all of you for joining us today. As I mentioned at the beginning of the webinar, we're going to post these slides and recording of the presentation on our website, the HDIAC website at www.hdIAC.org. If you're interested in learning more about the IACs or perhaps yourself getting involved as a subject matter expert or presenter or just simply expanding your presence in our user community, please feel free to reach out directly. We're going to put some contact information on the left-hand side of the screen, and you can get us that way. In addition to these monthly webinars, we also offer bi-monthly podcasts, bi-weekly email digests, and those include the latest scientific and technical news that may be of interest to you. If you register to become a member of our website, you can opt in for announcements about the podcast, the webinars, as well as receive the digest. And we also offer a free technical inquiry service where we can provide up to 4 hours of technical consulting and provide a nice little report to answer your question. And then we can also perform customer-funded work in any of our technical areas via our pre-competed contract vehicle, and that's something we really like to do. So again, that contact information is included in the chat box you'll see on the left-hand side of the screen. I'd also like to remind all of you to please be sure to join us on 18 December for our next webinar titled Biostasis. Some of you will recall that this one was going to be given around the election time at the beginning of last month, and we had to postpone it. And this one deals with the Defense Advanced Research Projects Agency and some work they're doing to extend the golden hour after a traumatic injury. The statistics have shown that survivability goes way up if you can get to a trauma center within an hour. And Dr. Tristan McClure-Begley from DARPA is going to talk to us about that on the 18th. So please join us for that. And then again, thank you very much for your time today. Steve, thank you for your time. Like us, follow us on social media as well. And you have all my best regards, and I hope that each of you stays well. Thank you.